Greetings and welcome to the Protein Synthesis Lab. This is currently set up in my kitchen because we are doing distance learning, and today we're going to learn about DNA replication and protein synthesis. And there could be some cameos between Artemis and Georgie. Not sure. This is Artemis. And my son Scott is holding the camera, and we'll be learning all of this great stuff about protein synthesis and DNA replication. Okay, so this piece of paper has a picture of a cell on it, and I've got my nucleus, ribosomes over here, and this is all of this good stuff in my cytoplasm. Right now, I will show you what I have for my DNA. This is my template. It's on, so we can differentiate between the DNA and RNA. I've placed my DNA on red ribbon, and there's a little bit of Velcro on the back. This is my template version. So I'll put it on the first. And then what we have here are these different colors. You can see there's 4 different colors here. We have green, red, yellow, and then there's some orange over here, or I could have done it here, green, red. The yellows, unfortunately, were not the correct size in this set, but I didn't realize, and orange. Okay, so this combination right here, these 4 colors, is everything that your body needs to make DNA. So this is the template strand, and there's a different combination all the way across of these little guys. I think they're called nucleotides. And then what this is a complement strand, because each of these little nucleotides has a complementary one that it likes to hang out with more than any other. So with just to get the right names on them, the green one is an adenine, and it loves to be with the yellow one, which is the thymine, I think.
And so this, am I allowed to ask questions?
Sure.
Is this in the strand of DNA, like the circular spinny thing? That's...
So, yep, and that's what it's going to become. So
and then they're connected because they're, I guess, like opposite each other, right?
Well, they're complements, so they stick together with a hydrogen bond.
Yeah.
Hydrogen bonds between these two pairs. Coming back to you yet?
Nope, but
okay.
I'm doing my best.
All right. Okay, so we've set up the complement pair, so the green and the yellow, they like to hang out. And the red and the orange, they like to hang out, or their names are, the green is adenine, the yellow is thymine, the orange is cytosine, and the red is guanine. The other thing I wanted to point out, because I don't think I did that yet, is that the way I've set these beads up, the greens, so the adenine and the red, which is the guanine, are 2 ring base pairs. So I've actually put 2 beads, one on top of the other. And then it's just unfortunate that just pretend that all 3 of those little yellows are just one yellow. And then like the orange one, they're just one flat ring base called pyrimidine, sorry. Okay, so on to the next thing. These 4 base codes make up everything to communicate protein synthesis, DNA replication. So basically everything in our body, everything in our cells is made out of protein. So these 4 things and the combinations of them makes everything in your body. So those 4 things creates everything.
Is it kind of like the 0 and the 1 for binary with computers?
Sure, except for there's 4.
Yeah, because I guess God couldn't quite... Use only 2. Sorry.
Please don't bring God.
I won't bring God into your DNA presentation. I'm sorry.
Get me marks off.
It just sounds like binary with a couple extra steps.
Okay, so yeah, and it's coding. That's basic, like they're called base codes. So yes, essentially that is what we're looking at. Is there's repetitive code, and things can get made out of the different combinations that they create, that they're made. So what I have then here is, this is the template strand. This is the complementary strand. So they each have their little partner that they like to hang out with. And then what I have done is...
Sorry, template and complement. I kind of wasn't focused on that. My bad.
Yeah. Oh, you didn't. Okay.
Well, I saw it now, but... in the moment. I was looking at the
Oh,
cat.
yes, and then they're together on the strip of... So what this happens is, this is now the alpha DNA helix thing that you were talking about earlier, that twists like that.
Yeah.
Okay? And that is found in the nucleus and only ever in the... Maybe I'll move that a little bit to the side.
Yeah, we could.
So it's only ever in the nucleus, but because of this really heavy Velcro strip, it won't stay folded. But anyway, we'll just pretend it's folded, and we'll pretend that it stays in the nucleus.
Yep.
All right. So, so the DNA is contains all the genetic code, and which is essentially the blueprint to create everything that you need for your body to duplicate and survive. And so the cells will need everything in this, but then how does it?
No? Okay,
No.
sorry, I'm just a bit strapped right now.
Okay. All right. Okay, so it makes protein is very important. This is all about how we're going to replicate this. And so basically there's some amino acids that come along and say, hey, we need your information and we're going to copy it over. And that's where the messenger RNA comes in. So what the messenger RNA does is it a single amino acid's added to this, and then it starts to okay, you need 20 amino acids to create all the proteins that are in your body. 12 of them are natural in your body. We can make 12 of them in our body, and then 8 of them we have to get from something that we eat. And one of my little nucleotides just fell off. Okay, anyway, so there are different kinds of codons that start and stop the replication and transcription process. And these we will go through when I'm showing you actually what happens in there. So the DNA and the RNA have a similar structure in their backbone. They're made with alternating sugar and similar phosphate and sugar. They alternate, excuse me, form a backbone. And in this one, you'll notice the colors are a little different. There's no, okay, so there's still adenine. And instead of the cytosine, there is a uracil here. So instead of the yellow, there's white. So that's why oops, I'm losing another one. Oh, this is going to be problematic. Okay, anyway, they're supposed to be staying on. Okay, anyway, so that's why you'll see white. So in the RNA, they have a few different things. And the other thing is about RNA is that it's a single strand. It doesn't join up with anything, but that's kind of a good thing because then it can easily get out of the nucleus because it's a single strand. And it doesn't... Where is she?
Who?
Georgie.
I don't know, but it's not... You're in the middle of a presentation. I don't think you're... Oh. I guess she got in my room. That's my bad.
Okay. This is so bad. This is so bad.
Just breathe. Take a quick breath.
Okay.
And just reset. Continue.
Okay, so the messenger RNA, these guys, what happens is... I'm going to just take that guy off. Because what happens is the DNA kind of starts to separate. I keep using kind of, but it's essentially what happens. So it unfolds a little bit, separates out, and then as it's separating, and kind of like that, the mRNA comes in and says, hey, I'll take some of that information.
So it's like bread and bologna.
So that happens. And it never, so there's
Oh,
different...
wait. No, it just...
There's different, yeah, it just comes alongside and there's different codons that it reads. Grabs the information, carries it over to the ribosome, goes through the ribosome with the different information on it, and the ribosome forms little tRNAs. We've got some sample ones here. This one would be like a toonie. It grabs that because it finds a UAC combination here. So green, white, and red would be the UAC. So we take the toonie off and goes there. And actually I think that's over there, and it brings it along here. So then it forms, we put the toonie there. Anyway, there's all of these guys, and some of this did not get communicated properly. I don't know if I didn't copy things properly or what happened, but we have all of these lovely transfer RNA. So this one's the messenger RNA, the strip of the black.
Should I
yeah, sure, whatever, doesn't matter. Okay, so these are just sample colors, and these actually are probably more in the way than they need to be. Okay, so some of this these are all the transfer RNA. So what happens is these guys come through. Okay, so the messenger RNA grabs, say, he finds out information. Oh, I need a toonie. So he comes through ribosome, this, and then the toonie goes, where was it here? Where's the toonie? The green, white, and red. So green, white, and red section here. And then that one goes off because these are all supposed to be in the cytoplasm. We just don't have enough space on the table. And he goes off and he grabs another one and comes back, and he's here with, I'm supposed to have another toonie on that. Anyway, we can set something like that. Anyway, so.
We couldn't find another toonie. Unfortunate.
I'm sure it's around here somewhere. Things have gotten a little away from me. Okay, anyway, so what happens is each of these things, oh, okay, and then this one, unfortunately, I think I did something wrong, but this is, say, like a toothpaste cap. So that's for the red, white, and orange one. Which I think I ended up doing something wrong because
there's no red, white, and orange.
Yes, so I'm not sure. So I'm not doing something properly. So we're just going to put this one over to the side.
What if
red, white,
you just
and
took
orange.
an orange and put it like right there?
Oh, actually, you're okay. There's no Velcro, but sure. No, they have to be in a row.
Like red, white, and then orange?
Yeah, like, okay, so if I did this, grab the white.
Oh, if you had another white in there, then you could make that
or actually,
the cap.
I could just put a red here. Red, white, and orange. So red, white, and orange. Okay, so then toothpaste cap goes there. Good catch, Scott. Well done. Okay, so then that one, where did it go? So it goes, finds another one, stops over there. Okay, so put more of there on. Okay, and then, so all of these have, okay, so for some reason they're not.
Also, don't sit down. There's a cat in your chair.
Okay. All right. So then, so each of these combinations have to have items out of the cytoplasm that these are tRNA. So they are grabbing the stuff from the cytoplasm, and it's interpreted by the messenger RNA. So it's a combination. They work together as the RNA, and all of these things, I don't know if I'm actually going to get this part done properly, but it's basically supposed to be there's a combination of these items. I don't think we even needed that one. Like that. So that all becomes like, oh, and sorry. And then I think our blue pen was supposed to end it. Like it's an end codon. So then this gene information has been copied over here or transcripted in the nucleus, brought out, and then okay, anyway, transcription is this part where it grabs the information out of the nucleus from the DNA, and then the translation of it is when the mRNA codes with the tRNAs, and then all of the information is lined up. Then we have a stop codon that comes in and says, hey, that's it, that one's over, that one's done, and it ships it all off, and like that. Okay, so that's all, that one's done, that's done, but it stays in a loop that can be used by your body. Okay, so I don't know if I can keep going. This just seems like it's taking way too long. Okay, so Okay, so yeah, so it actually gets transported in that way. So then the mutations that I want to talk about is there's a couple of things that happen where there's mutations. A very good example I would like to bring up would be smoking, because smoking is carcinogenic and it makes your cells reproduce inaccurate information. So that would translate in this situation like, hey, we want to have this information communicated, and oops, here, let's get the extras off that. And this guy says, hey, okay, I can handle that, but instead of the white one, it's a blue one, and says, hey, that's a smoker. He's got a little bit of damage there, so I'm going to take that and communicate that to the rest of it because I can't read what it really is. So it comes over and that information gets communicated. Now the type of mutation, it could be a mistake in the DNA. It could be a mistake in the communication between them. So the different things that we call them, environmental factors would be a carcinogen, smoking, chewing tobacco, radiation, UV rays. Those are things that are environmental. And then the point mutations is one type of single base mutation. That would be an example. I just used one small little tile just to give an idea. It would just be one small tiny little mutation, and it gets substituted for another. And then it could possibly alter a single amino acid in the chain, but not always. So it's just like one tiny little thing that's different. But if you get enough tiny little things, then that can cause trouble. A frame shift mutation is when there's an addition or deletion of a single base. So that would be more than just one of these guys. And it changes the whole reading of the codon so that every amino acid afterwards will be giving the wrong information, but it's the new information. So it's the latest information that's being communicated through. So then that will absolutely alter the gene expression. And so there can be silent mutations, and that can be things that occur that are not in the non-coding sections of your DNA. Mutations that occur or have changes in DNA bases, but the amino acids stay the same, like if there's a small tiny little thing like that. And an example of that would be with your teeth. There's some multiple codes for ARG, and so there is a possibility that there could be a mutation in that, and it would still code for arginine, which would be for what is that in your Red Bull, do you think? Arginine? Do you think that's, because no. I'm sure the amount of sugar and caffeine alone would be not healthy, but we're not talking about not healthy, we're talking about gene mutation here. Okay, and so it would still code, so it's possible the amino acids can still code for a full thing like arginine, but there could be things going on inside. So there's a potential there for something to go wrong. And then there's missense mutations that are the way the protein is shaped. So it just works less effectively or maybe doesn't work at all. So I think that's pretty much the way we've dealt with the protein synthesis. We've dealt with the strands and how they have their little partners they always like to be set up with. Then we did the translation and transcription and mutations, and I believe that is it. I really hope that this is that and understood. Scott, do you have any questions?
Nope.
Alrighty. Okay. Thank you very much, and appreciate you listening to this.